Okay, today we are going to be discussing epigenetics, which literally means on or near genetics. That's because epigenetics does not deal with changes to your genetic information, such as point mutations or frame shifts. Epigenetics solely deals with ways that the environment affects how your body can read your genetic information. So in your somatic cells, you typically have, on average, 6 feet of DNA. That is a lot of DNA to store in a somatic cell. So what the body does is wraps that DNA around proteins called histones, which means that now you have 6 feet of DNA reduced to less than 6 feet of DNA because it's basically been coiled. It's been folded like tying up a rope on a boat or some such. So this changes how well the body can read that DNA. If the body chooses to methylate the DNA or add methyl groups to that DNA structure, so same set of base pairs, but now a methyl group has been added, the DNA gets wrapped very tightly around these histones, and it's harder for the body to read it. If it's demethylated, then the DNA is loosely wrapped around the histones, and it's easier for the body to read it, because now the RNA can access the DNA, transcribe it, and code for proteins. So Professor Meany from McGill University did a study called Maternal Care Associated with Methylation in 2006. He found that affection during child rearing among rats can methylate or demethylate a gene that codes for estrogen receptors. So let's say that you have, this is a high affection mother, and in the process of being affectionate with its child, that child will also become a high affection rat if it does become a parent, and that cycle will continue with environmental influences demethylating a gene that codes for an estrogen receptor. However, if the mother is low affection, then it will methylate in its child the gene that codes for an estrogen receptor. So to be clear, this isn't a trait that it's passing on, but as this rat child is being brought up, its gene that codes for an estrogen receptor is being methylated more and more. It's getting harder and harder to read, so it's not ending up being read, and it becomes an unaffectionate rat child, and eventually an unaffectionate rat mother if it has children. Now, the interesting thing is, if a high affection mother rat fosters a low affection mother's child, then that low affection child will become a high affection child, and it will have its methylated DNA over here become demethylated. So because it's been demethylated, the estrogen receptor can now be transcribed for, it can be coded for, and it will become an affectionate rat. Now, Professor Seif also did studies in terms of the heritability of emotional conditions in his study Lamarck Revisited from 2014. He exposed a generation of mice to acetophenone, then shocked them with electricity. This caused the gene that coded for an olfactory receptor to become demethylated. That demethylated gene was passed on to the children of those mice that were exposed to acetophenone and then shocked, so that those children were afraid of the smell of acetophenone even though they'd never directly experienced the shock that comes afterwards and had no idea that that was going to happen. Then finally we have Professor Yehuda's study, Holocaust Exposure and FKBP5 Methylation from 2015. This one's a little complicated, so bear with me. Essentially, Holocaust survivors had a very highly methylated FKBP5 gene. So just to recap, because a gene is supermethylated, that means it's not really being read or transcribed for. The FKBP5 gene is responsible for modulating emotional reactions, so chances are, if you're in a life or death situation, you don't want to be modulating your reactions too much, so it got methylated. You might assume that their children also had a very highly methylated FKBP5 gene, when in fact, they had a very, very demethylated FKBP5 gene, a very loose wrapping of the DNA around the histones, looser than the average population, which essentially meant that not only were they more capable of dealing with stressful reactions in a deliberative manner than their parents, but then the population as a whole, those people that hadn't descended from Holocaust survivors. So because the FKBP5 gene was so expressed, sometimes there were issues in terms of these people being more prone to dissociation, but it also meant that they were more capable of staying cool under pressure and reasonably reacting to very unreasonable situations. So just to recap, the environment can control how much your body methylates or demethylates a gene. If a gene is highly methylated, it cannot be transcribed for and thus won't be expressed. If a gene is demethylated, it will become more expressed because it can be transcribed for, but it's that same gene. The base pairs aren't changing, the gene is still there, it's not being deleted or mutated. But the environment can change it, and those environmental changes can be passed on from generation to generation, which can have critical implications for survival. Thank you very much, and I hope you have a great day.